In the previous video, we investigated osmosis in plant tissue. But what is happening inside the cell? In this video, we'll be looking at osmosis in plants and in mammals. If a plant cell is placed in an isotonic solution, where the concentration of the solute outside the cell is equal to the internal concentration, the cell will stay normal. While if a plant cell is placed in a hypertonic solution, water will move inside the cell by osmosis. This causes the vacuole to swell, pressing the cytoplasm against the cell wall. As a result, the pressure builds up and causes the cell to swell and become very turgid. Whereas if a plant cell is placed in a hypertonic solution, water will leave the cell by osmosis. The cell becomes flaccid, as there's no pressure on the cell wall, causing the vacuole and the cytoplasm to shrink, and eventually the cell membrane pulls away from the cell wall, and the cell is now plasmolyzed. Unlike plant cells, animal cells do not have a cell wall. So let's see what happens in this case. We will take red blood cells as an example. Red blood cells are transported within the blood plasma. If the solution outside the cell becomes much more diluted than the cell content, water moves in by osmosis. The cell then swells and might burst. While if the solution outside the cell becomes much more concentrated than the cell content, water moves out the cell by osmosis. The cytoplasm becomes very concentrated and the cell shrivels up and can no longer survive. To prevent either situations, our bodies have a control mechanism to make sure that the cells are continuously surrounded by blood plasma, which is isotonic, to maintain the cell's biconcave shape.